So, welcome back, everybody, to the podcast of the InnCoCells project, making innovative high-value cosmetics from plants and plant cells. We're delighted to be here in Helsinki for the third project meeting, and the second one where everyone has been able to meet face-to-face, largely free of COVID restrictions this time. And in this edition, we welcome Dr. Gianfranco Diretti from Enea in Rome. Hello, Gianfranco.
Hello.
Very nice to have you here. Perhaps you could tell us a bit about, for our listeners, about Enea as an organisation and your history working there.
Sure. Enea is the National Agency for New Technologies, Energy and Sustainable Development. We have different centers that are distributed along the countries, and more specifically, we are working in Rome. And we have a series of activities, and we place like the third in research institution in Italy in terms of size and number of personnel.
So how many personnel are we talking?
Overall, something like 2,500.
So your department, how many people work in your particular department?
We are in the Department of Sustainability, and we cover several activities. More specifically, we are in the Biotechnology and Bioindustry division and the Laboratory of Biotechnologies that is taking care of all the activities that are running in InnCoCells.
Okay, so that's actually was going to be the next thing I was going to bring up. So what would you say is the key role of Enea in the InnCoCells project?
Enea is involved in two work packages. The main role of Enea is in work package 5. We have the leadership of the work package, and in the frame of this work package, we are going to perform a quite large metabolomics activity aimed to profile all the samples that have been shown to have a cosmetic interest.
This is maybe something we should explain for our listeners who aren't specialists. Metabolomics is basically where you take a sample, in this case would be a plant matter of some kind, and you look at all the small molecules which you can find in it. And essentially what we're doing is trying to find these small molecules which we can use as cosmetic ingredients. So when Gianfranco mentions metabolomics, he's talking about screening all these plant tissues and making sure that we can find the key ingredients, which are small molecules, which we can then use to develop into cosmetics. Would you say that's a fairly accurate layman's summary of what we're talking about?
Yeah, basically metabolomics allows in one shot to detect the highest number of compounds or chemical compounds that can be found in a sample. And in the frame of InnCoCells, the concept is just to perform, first of all, a series of bioactivities on all the samples that will subject it to this bioprospecting activity, and ones that are going to show any positive effect with respect to the assays under investigation will be then subjected to a deep chemical profiling by metabolomics in order to identify the compounds responsible for that activities.
Okay, so is this something that's like a personal interest to you? Have you always been interested in this particular aspect of biotechnology, or is the InnCoCells project a new extension of your former work?
Well, the metabolomics and in general apply to biotechnology is an interesting combo, let's say, that is ongoing since several years. The novelty in the case of InnCoCells that we are applying this kind of strategy to the cosmetic field.
So that's very interesting to hear. So, tell us, is this something that's always interested you? I mean, were you fascinated in it as a child, or how did you come to be involved in biotechnology as a topic?
Well, since education, I'm a plant biotechnologist. I was always fascinated by plants, and especially by the large set of compounds that a plant can synthesize. Of course, compounds can belong to the, let's say, primary metabolism, which comprise all the molecules that are necessary for the life of a cell, and on the other side, the secondary metabolites ones that are the one that are providing an evolutionary advantage for a cell. And from this point of view, the exploitation of the secondary metabolism of the different plants is a very fascinating field, and I'm working on that since 10 years with different purposes.
And before that, was this right at the beginning of your career, or were you in a previously working on a different topic and you then branched into plant secondary metabolism?
Yeah, for instance, we worked on a secondary metabolism with nutrition purposes. More in detail, we have worked for several years on vitamin A metabolism, since vitamin A deficiency is a quite serious problems, health-related problems in a series of poor countries. And we work on the metabolism of vitamin A, that is that regard the pathway of carotenoids. And in this frame, we engineering tomato and potatoes, especially potato is devoid of carotenoids and is one of the 4 main crops in terms of distribution and consumption worldwide. And we generated by metabolic engineering approach crops that were enriched in total carotenoids and specifically beta-carotene.
It's very interesting because I've worked with a couple of groups who do similar work in corn, and obviously, I mean, you'll be very well aware of the Golden Rice Project and how that's worked out. Do you foresee the golden potato becoming mired down in similar problems, or do you think it's now easier to find that path to commercialization?
Well, you can think about the very quite difficult and long path that is still ongoing for the golden rice. Golden rice has been produced formally with respect to our golden potato. Now it's after open field trials and a series of let's say ethic problems and so on. A large distribution of the golden rice is still missing. So we performed a series of studies to investigate the effects due to the metabolic engineering attempt. We performed nutritional studies, so that's proving that these potatoes can represent a valuable source for vitamin A. But to be honest, we are still far from a large distribution.
So, do you see any parallels between the regulatory problems that you've had with golden potato, and the regulatory work that's going on in the InnCoCells project, where we're going to have the cosmetic ingredients that we're producing in plants approved for use in humans?
Well, for sure, the regulation is different. I mean, in both cases, there are restrictions and limitations, and a series of issues that you have to take into account. But while the GMO issue, if we are talking about edible stuffs, is still a problem. On the other side, I mean, as far as I can see, in the case of the cosmetic ingredients, we have to comply, of course, with a series of regulation and European and worldwide level, but it's not so tight like in the case of the edible samples.
And this is because it's a topical application rather than an ingestion of the product.
Yeah.
So for our listeners, we are currently sitting on an island in Helsinki at a very beautiful hotel where the conference is being held. And we've been listening to the most recent updates from the project partners. And Gianfranco, I know you're not giving your presentation until tomorrow, so I'm not asking you to give any spoilers, but of course our listeners won't know you're giving the presentation tomorrow because by the time this is published, the conference will have been over. So just for the podcast listeners, have you got anything you can tell us about the progress in the project from your side and how you think it's going, especially, the work that NAO is doing?
Sure. For the NAO activities in InnCoCells, we have set up optimized conditions to obtain the in vitro cell cultures of two species. One that is quite famous is the purple basil, and another one, an Asian plant with a series of compounds of interest for the cosmetic field, which is called perilla. And we optimize all the conditions to obtain the valuable amount of biomass that are now in the phase of the screening in terms of bioassays as well as chemical profiling. As well, we have developed a vertical farming platform based on the use of hydroponic conditions coupled to LED light system in order to stimulate the production of secondary metabolites in microgreen and exotic species from the Zingiberaceae family, like a wild ginger species, which is called Zingiber zerumbet, and another one which is called Campharia galanga. And now we are in the phase to ship all the samples to the partners that are performing the bioassays, and also in the phase of the chemical profiling. From this point of view, all the procedures for the liquid chromatography and GC chromatography coupled to mass spectrometry have been set up.
Excellent. So you're happy with progress, and you can see this ending up being a successful project?
Yeah, for sure. And then we have a last part for metabolic engineering in tomato, and we were able to, by using CRISPR-Cas9 technology, to edit finally the two genes involved in the accumulation of anti-negative components like glycoalkaloids and allergen. And we are now in the phase to profile and to screen also this sample.
Excellent. So what would you like to see this project ultimately achieve, not only in terms of the stated objectives of the project, but on a personal level, and also for ENEA? What would you like to see the outcome of the project being?
Well, this is for ENEA is a very good opportunity being part of such an important consortium, and also in a field which is having a growing interest during the last year. And also another nice point of view for ENEA, and that we were able to combine a series of colleagues that are having different expertise. So we are basically covering for the all the InnCoCells activity, people with in vitro culture expertise, agronomists for especially for the vertical farming part, as well biotechnologists to do metabolic engineering, as well plant metabolomics.
Excellent. So do you think this will actually direct some of the future research ideas and strategies at ENEA?
For sure. We are, it is from a side helping higher coordination and cooperation for the personnel that are involved in the project, as well by combining the different expertise. I'm quite sure that it will help to develop future strategies and opportunities in different scientific fields.
That's absolutely fascinating to hear your input to the project, Gianfranco, and it's been a great pleasure having you here on the podcast of the InnCoCells project. Thank you very much to our listeners for joining us again, and next time we'll be joined by Frederick Borgard from Plant Advanced Technologies, and we'll be hearing more from him in the next instalment. So again, thank you everyone for listening, and join us next time, and we'll enjoy your company soon. Thank you very much.